Imagine a bustling city. Buildings stand tall, each with walls that define their boundaries and protect what's inside. Cells, the fundamental units of life, have their own versions of walls and fences, cell walls and cell membranes. These structures are essential for life as we know it. They provide structural support, act as barriers, and regulate the movement of molecules in and out of the cell. Cell walls are like the sturdy outer walls of a building, providing rigidity and protection. They are found in plants, fungi, and bacteria, but not in animals or protozoa. Cell membranes, on the other hand, are like the security checkpoints, controlling what enters and leaves the cell. They are present in all living cells, both with and without cell walls. Without them, cells would be vulnerable to damage, unable to maintain their shape, and incapable of regulating their internal environment. From the towering trees to the smallest bacteria, cell walls and membranes play a crucial role in the survival and function of all living things. Plant cells are easily recognizable by their rigid cell walls. These walls are primarily made up of cellulose, a complex carbohydrate that forms long, strong fibers. Imagine cellulose as the steel beams of a building, providing the structural support that allows plants to stand upright and reach for the sky. The cell wall in plants is not just about strength. It also plays a crucial role in protecting the cell from bursting when it takes in water. Think of a balloon being inflated. Without a strong outer layer, it would simply pop. Similarly, the cell wall prevents plant cells from experiencing a similar fate. It has tiny channels called plasmodesmata that connect neighboring cells. These channels allow for the exchange of nutrients, water, and signaling molecules between cells, ensuring coordinated growth and development. Fungi, unlike plants, don't rely on cellulose for their structural integrity. Instead, their cell walls are primarily composed of chitin, a tough, flexible material also found in the exoskeletons of insects and crustaceans. Think of chitin as a chainmail armor, providing protection while still allowing for flexibility. The chitin-rich cell walls of fungi serve a similar purpose to those in plants. They provide structural support, protect the cell from bursting, and act as a barrier against pathogens. Chitin is highly resistant to degradation by bacteria and enzymes, making fungal cell walls particularly tough and durable. This resilience is crucial for fungi, which often thrive in harsh environments. Prokaryotes, which include bacteria and archaea, are single-celled organisms that lack a nucleus and other membrane-bound organelles. Despite their simplicity, they possess a unique cell wall structure that is crucial for their survival. Bacterial cell walls are primarily composed of peptidoglycan, a complex polymer made up of sugars and amino acids. Imagine peptidoglycan as a tightly woven fabric, forming a strong mesh-like structure around the bacterial cell. The peptidoglycan layer provides structural support and protection, preventing the bacterial cell from bursting due to osmotic pressure. The structure of the peptidoglycan layer can differ between bacterial species, leading to the classification of bacteria as either gram-positive or gram-negative. Unlike cell walls, which are only present in certain organisms, cell membranes are a universal feature of all living cells. They act as a thin, flexible barrier that separates the cell's internal environment from the outside world. Think of the cell membrane as a delicate but effective fence, controlling the flow of molecules in and out of the cell. The cell membrane is composed primarily of phospholipids, a special type of fat molecule with a hydrophilic or water-loving head and two hydrophobic or water-fearing tails. These phospholipids arrange themselves in a double layer, with the hydrophilic heads facing outward towards the watery environments inside and outside the cell, and the hydrophobic tails sandwiched in between. This unique arrangement creates a selectively permeable barrier that allows certain molecules to pass through while restricting others. Small, uncharged molecules like oxygen and carbon dioxide can diffuse freely across the membrane, while larger molecules like glucose and ions require the assistance of specialized transport proteins. The cell membrane is not just a passive barrier, it's a dynamic structure that is constantly changing and adapting to the needs of the cell. Phospholipids are the stars of the cell membrane show. These fascinating molecules are the primary building blocks of this vital cellular structure. They are uniquely suited for this role due to their amphipathic nature, meaning they have both hydrophilic or water-loving and hydrophobic or water-fearing regions. Imagine a phospholipid molecule as a lollipop with two sticks. The lollipop head, containing a phosphate group, is hydrophilic and attracts water. The two sticks, representing fatty acid chains, are hydrophobic and repel water. This dual nature drives the self-assembly of phospholipids into a double layer when in an aqueous environment. In this bilayer, the hydrophilic heads face outwards, interacting with the watery environments inside and outside the cell. Meanwhile, the hydrophobic tails tuck themselves inwards away from the water, creating a hydrophobic core. This arrangement forms a stable barrier that effectively separates the cell's internal environment from its surroundings. The fluidity of the cell membrane is also regulated by the properties of the fatty acid tails in phospholipids. While phospholipids provide the structural framework of the cell membrane, proteins are the workhorses that carry out many of its essential functions. These diverse molecules are embedded within the phospholipid bilayer, acting as gatekeepers, transporters, communicators, and more. Imagine proteins as specialized personnel stationed within the cell membrane's security checkpoint. Some proteins, known as transport proteins, act as channels or carriers, facilitating the movement of specific molecules across the membrane. These molecules could be nutrients entering the cell or waste products exiting. Other proteins function as receptors, receiving signals from the external environment and relaying them to the cell's interior. This communication is crucial for cells to respond to changes in their surroundings, such as the presence of hormones or toxins. Enzymes, another type of protein, can be found embedded within the cell membrane, catalyzing various chemical reactions necessary for cell function. The types and quantities of proteins present in the cell membrane vary, depending on the cell type and its function. Carbohydrates, often overlooked in the cell membrane's bustling environment, play a crucial role in cell recognition and communication. These molecules are typically found attached to proteins or lipids on the outer surface of the cell membrane, acting like personalized ID tags. Imagine carbohydrates as flags or banners displayed on the cell's exterior, signaling its identity to other cells. These carbohydrate markers are recognized by specific proteins on other cells, allowing for cell-to-cell recognition and communication. This recognition process is essential for various biological processes, including immune responses, tissue development, and fertilization. For instance, immune cells rely on carbohydrate markers to distinguish between the body's own cells and foreign invaders like bacteria or viruses. Carbohydrates on the cell membrane also play a role in cell signaling. They can act as receptors for signaling molecules, triggering specific responses within the cell. The cell membrane's selective permeability is crucial for maintaining a stable internal environment, also known as homeostasis. To achieve this balance, cells employ various mechanisms to transport molecules across their membranes. Imagine the cell membrane as a busy border crossing, with molecules constantly moving in and out. Some molecules, like oxygen and carbon dioxide, can cross the membrane freely through diffusion, moving from an area of high concentration to an area of low concentration. However, many essential molecules, such as glucose and ions, cannot simply diffuse across the membrane. They require the assistance of transport proteins, which act like specialized vehicles, facilitating their movement. This process, known as facilitated diffusion, still relies on the concentration gradient, with molecules moving from high to low concentration. Sometimes, cells need to move molecules against their concentration gradient, from an area of low concentration to an area of high concentration. This uphill battle requires energy and is achieved through active transport. Cell walls and membranes are fundamental structures that play vital roles in the survival and function of all living organisms. They provide structural support, act as barriers, regulate transport, and facilitate communication between cells. Cell walls, found in plants, fungi, and prokaryotes, provide rigidity, protection, and shape to cells. The composition of these walls varies depending on the organism, reflecting their specific needs and adaptations. Cell membranes, present in all living cells, act as selectively permeable barriers that regulate the passage of molecules in and out of the cell. They are composed primarily of phospholipids, proteins, and carbohydrates, each contributing to the membrane's diverse functions. These cellular boundaries are essential for maintaining a stable internal environment, facilitating communication, and ensuring the overall health and survival of organisms. Understanding these structures provides a greater appreciation for the intricate workings of life and the interconnectedness of all living things.